Hello, my name is Kaylee Hazel, and I'm going to present this article for our lab assignment for Biology 2440. And we were asked to find an article that covers our protein that we were given, and my protein is DNA polymerase. Again, the article was DPB2 integrates the leading strand DNA polymerase into the eukaryotic replosome. The article was authored in the United Kingdom. The authors are a part of the Patterson Institute for Cancer Research and the University of Manchester in the United Kingdom. The authors included Dr. Sengupta, Dr. Durson, Dr. Piccoli, and Dr. Labib. Some background on this study: the eukaryotic replosome is a critical determinant of the genome integrity with a complex structure that remains poorly characterized. They were wanting to find some more information because the eukaryotic DNA structure is still a pretty good mystery for most scientists today. There's still a lot that we can learn and study on that. The researchers in this study wanted to determine how the helicase is linked to the DNA polymerase. This synthesizes the leading strand at replication forks and is an important focus for regulation of DNA replication. In this article, the researchers covered a study that they did on budding yeast. The yeast the researchers found has a compound in it called DPB2, which is actually found in all eukaryotic DNA replication processes. They found that it is essential for the replication initiation. The DNA polymerase doesn't just spontaneously come out and say, hey, I'm going to replicate here. The DPB2 is actually needed to initiate that first instance of replication. They found that the compound, including DPB2, is required for the initiation of the assembly of the helicase, which is the outside covering. Researchers found that 2 of the 3 DNA polymerase during replication are actually going to be working and active at one time, and they're working together to replicate the DNA itself. What I found interesting, when I'm probably going to be using some in my paper, was that that there are 3 DNA polymerase compounds, and while replication is occurring, 2 of those compounds are working together to mediate synthesis of the leading and lagging strands, while the third complex serves as a backup. That is important to avoid gaps on the lagging strand and maintains a high rate of fork progression. This makes sure that the DNA replicates properly, because we all know when DNA doesn't copy itself perfectly, we have major issues. Though DNA polymerase is an essential part of the DNA replication, there are other enzymes and compounds needed to initiate this process, which is probably going to help me a lot in writing my paper for the rest of the semester. I hope that you are having a wonderful day. If you have any questions over the article or anything I've said in this presentation, please feel free to contact me. Again, hope you're having a wonderful day. Thank you.